Okay guys, here we go. Introduction to the immune system. Y'all, let me just tell you, this is fascinating, okay? Really, really interesting. Once you understand how efficient and how complex our bodies are, especially with this immune system, it's just nothing short of amazing. So, with that being said, let's look at here. We basically have 3 lines of defense against pathogens. The first line, you already know. This is your barriers, your surface barriers, cutaneous and your mucous membranes. They're going to physically block entry of pathogens into the body. Then you have your second and third line of defense. Now this is where you have the responses of cells and proteins that make up 2 types of immunity: innate immunity and adaptive immunity. So that's kind of where we're going with this second and third line. So let's look at this. Surface barriers, first line defense against any threat to the body. These are covering, skin, mucous membranes, also the products they secrete. The skin and mucosa have a continuous physical barrier. Your skin is resistant to mechanical stress because of all the epithelial cells and that keratin, hard protein. Your sebaceous glands secrete sebum, which gives us that slightly acidic pH mantle, which deters bacteria from growing on the skin. All of this you learned in 201. Remember, your mucous membranes line all passageways that open to the outside: respiratory, GI, urinary, as well as reproductive. This is where we lack keratin, so we're less resistant to mechanical abrasion, but they secrete mucus, which is a really sticky substance. That mucus traps pathogens and it protects the underlying cells from any chemicals and any more trauma. It also, the mucous membranes of your stomach secrete stomach acid, which of course kills any pathogens that come into that location. Alright, so let's look at our 2 types of immunity. This is the second and third line. And this basically differs in how they respond to the different pathogens and the form of cell injury, basically. Alright, so the first one's called innate or non-specific immunity. And the deal here is it responds to all pathogens in the same way. Okay, now here are some components of that. Antimicrobial proteins in certain cells, they respond very quickly. They are the dominant response for the first 12 hours after exposure, either exposure as to a pathogen coming in or an injury. The cells and the proteins exist in the bloodstream, even in the absence of a stimulus. They're always circulating in the blood. This immunity does not have the capacity for immunological memory, so it's going to respond in the same way with a repeat exposure to a pathogen. So there is no memory. There are no antibodies that are going to be made with non-specific or innate immunity. All right, they don't have that capacity, lack that capacity. So they're going to respond the same way every time you're exposed to that same pathogen. Now, adaptive or specific immunity responds individually to unique glycoprotein markers called antigens. Now, we just talked about antigens. They identify a cell or molecule as a specific group. They give it cell ID, unique ID. All right. Now, this type responds a lot more slowly than innate because one must be exposed to that antigen in order for a response to be initiated. Sometimes we call this acquired immunity because it takes like 3 to 5 days to get a response together, but after that point it is a dominant response. Now here's the big difference. It does have that capacity for memory. So once it's exposed to an antigen, it remembers that and it makes specific lymphocytes and antibodies to fight that off the next time you're exposed to it. So this is a really fast and efficient way if you have a repeat exposure. Now there's 2 arms, so to speak, of the adaptive immunity. You have the cell-mediated and you have antibody-mediated. Now, something to look at. This type, adaptive, and the first type, the innate immunity, are not independently functioning arms of the immune system. Each type relies on each other. They work together. They respond to pathogens with a really highly integrated series of events we're going to look at within both parts. So they're not independent. They do work together, and it takes both types to adequately protect our body. All right, so Pathogens that are able to get through those surface barriers have to meet the second line defense, and that's going to be your cells and proteins of innate immunity. Now, there's two broad types of cells in innate immunity, phagocytic and non-phagocytic, and you know what phagocytic cells do. That's going to be your macrophages, your neutrophils, and your eosinophils. They, of course, are going to ingest particles by phagocytosis. Now, macrophages are activated by several different stimuli, certain molecules in the pathogen, chemicals secreted by the damaged cells, even signals from the adaptive immunity will elicit and activate these macrophages. Once they're activated, the local macrophages are generally the first cells to get to the injury site, and they're going to ingest other cells and any cell debris that needs to get out of the way. They also kill pathogens that they've ingested with chemicals. They can secrete hydrogen peroxide or hypochlorous acid, which is a very active part of bleach, so that tells you how powerful it is. They can kill their ingested pathogens by secreting these chemicals. They also have cytotoxic effects, so they can secrete substances onto the pathogens that are too big for them to ingest. They also can function as APCs, which are antigen-presenting cells. So these are cells that display parts of the pathogen, the antigens on the pathogens, and then they ingest their plasma membrane. The T cells are activated by the antigens and in turn can release substances that increase the activity of the macrophages. So this is basically a positive feedback. The macrophages release chemicals that activate the T cells. The T cells release chemicals that increase the activity of the macrophages. So they're going to go back and forth, which again is a positive feedback loop until we get this under control. Whether it's the injury or the infection spreading, whatever it is, you're going to work this way until it's under control. So the macrophages do a lot. All of these are important functions for the macrophages. Now, neutrophils, remember we've studied these. They really like to eat bacteria. Most numerous granulocyte we have. They are highly effective phagocytes. They kill the pathogens with chemicals like hydrogen peroxide and hypochlorous acid, same way the macrophages do. They ingest lots of different, but they really like to destroy bacteria. They do release contents of their granules onto these big pathogens to damage the membrane. They generally are residing in the blood, but they can be recruited by chemicals to different damaged areas. And then you have eosinophils, which are also phagocytes that migrate from the blood to wherever they're needed at. They like to respond to parasitic pathogens. These are parasitic worms we discussed. They cover the parasite, release the content of their granules. Remember, their granules are like lysozymes. They store those powerful enzymes and chemicals. The chemicals from those granules damage the parasite and either destroy it or make it easier for somebody else to come along and destroy. Okay. Then you have the non-phagocytic cells. These are your natural killer cells or your NK cells, your dendritic and your basophils. Your natural killer cells have a remarkable ability to recognize cancer cells and cells that are infected with certain types of viruses, in spite of the fact that they can't recognize specific antigens. So really interesting there. They can recognize cancer cells and virus-infected cells, but they can't recognize specific antigens. They are cytotoxic, so they release chemicals to destroy their target cell. They also secrete antimicrobial cytokines that really activate macrophages and enhance phagocytosis. Your dendritic cells function as antigen-presenting, so they ingest the antigen from the T cell, or they ingest it from the pathogen and they present it to the T cell. Now the T cell can determine whether that's self or foreign. The dendritic cell can't. The dendritic cell doesn't know if it's self or foreign, but the T cell can make that decision. And then you have the basophils, which are rare, but their granules have chemicals that mediate inflammation. So these are your inflammatory mediators, mainly located in the blood, although there is a specific type called mast cells that are located in the mucous membranes, and just like the regular, they have chemicals that can trigger inflammation. These are mainly involved when you have allergic responses. Okay, so this is going to be a lot of those histamine reactions with itching, eyes watering. Those things typically are going to be activated by the basophils or taken care of by the basophils. All right, we are actually going to stop there for now. We will move into antimicrobial and the complement system in the next video.